Hello, I'm Andrea Gomez Donart, one of the scientists at Sartorius in Albuquerque, New Mexico. Today I will be walking you through one of our posters presented this past August at CYTO, and this poster is titled An Optimized Multiplex Assay for Screening the Functional Capacity of Bispecific T-Cell Engager-Induced Killing using Advanced Flow Cytometry. Before I get into the assay setup and data generated for this presentation, for just a quick background on immune cell killing, we know that upon recognition of cancer or damaged cells, cytotoxic T-cell lymphocytes become activated and release effector proteins and cytokines during the immune response. Some of these proteins are responsible for killing the cancer or damage-causing cells. The ability to monitor this process may be beneficial in assessing the efficacy of small molecule biologics, which are intended for cell therapeutics. Here we've developed an advanced flow cytometry phenotype and function assay kit to be used in conjunction with our IQ3 platform, which identifies T cells, distinguishes them from target cells, assesses cell viability to monitor cell death or killing, determines expression of T cell surface markers for activation and exhaustion, and monitors the secretion of pro-inflammatory cytokine interferon gamma along with the pro-apoptotic protease granzyme B. In this poster, we demonstrate the killing of target cells by effector cells, which are activated by two different methods, either a bispecific T-cell engager or BiTE and CD3/CD28 Dynabeads. The data were acquired using the Sartorius IQ3 VBR. The IQ3 is an advanced flow cytometry platform that can be used for antibody screening and functional profiling. Its integrated Foresight software allows for fast data acquisition and analysis, as well as a dynamic visualization of results. The easy-to-use kits have pre-made templates which assist in easy, fast analysis and offer users a variety of visualization methods. Shown here is the experimental design used to demonstrate T-cell mediated killing. Briefly, Ramos cells, pre-encoded with blue-green encoding dye, were co-cultured with PBMCs from 3 different donors, and the PBMCs were activated by either an anti-CD3/CD19 positive BiTE or CD3/CD28 Dynabeads. Both the positive BiTE and Dynabeads were prepared at 6 different concentrations, including a 0 activation of media only, which demonstrates a dose response to the activation. In addition to the activation dose response in co-cultures, to demonstrate the effect of the treatments on single-cell cultures alone, the Ramos cells and cells from each donor were treated with the 2 positive activation methods at the top concentration, along with 2 negative activation treatments, which were media alone and an anti-CD3 beta-gal negative BiTE. The final effector to target ratio in the co-cultures was 5:1, and the co-culture plate was held in a humidified incubator at 37 Celsius for up to 72 hours. At 4, 24, 48, and 72 hours, a well-mixed 10 microliter aliquot from each well was transferred to a V-bottom assay plate and assessed using the IQ Human T-Cell Killing Kit. Here details the multiplex assay workflow, where targets are pre-encoded with an encoding dye and co-cultured with effector cells for a designated amount of time. At the desired time point, an aliquot of sample is transferred from the co-culture plate to an assay plate along with the cytokine capture beads and incubated for 60 minutes. There are 2 more 60-minute incubations following the addition of a cytokine detection reagent and an antibody detection reagent, after which the assay plate is washed and the samples are resuspended in a final 20 microliter volume. The sample data can be acquired on the IQ3. The total assay time for each time point is just over 3 hours, and the total acquisition time is about 20 minutes for a 96-well plate. The advantage here is that the same culture plate can be utilized for multiple time points across a time course of several days. This slide demonstrates the multiplex assay design. showing the various metrics determined from each individual well. The effectors and targets are differentiated from each other by the encoding dye, and live and dead cells are identified using a viability dye. Surface marker protein antibodies identify CD3 positive, CD8 positive cytotoxic, and CD3 positive, CD8 negative helper T cells. Also included are surface marker protein antibodies to indicate the T cells which are expressing CD25 and PD1, the activation and exhaustion markers. And lastly, secretion of interferon gamma and granzyme B is measured to demonstrate the inflammation and killing. This demonstrates the assay gating strategy, which is all preset in the Foresight assay template. Cells and beads are determined by forward scatter and side scatter. Singlet Q beads are then determined and separated out by brightness to differentiate and measure amounts of interferon gamma and granzyme B in each sample. Singlet cells are identified and able to be separated into targets and effectors, which can go further to live dead targets and live dead effectors. From the live effectors, we can determine the CD8 positive and the CD8 negative T cells, and from there, we can see which cells are expressing activation or exhaustion cell surface markers. The count of live targets and target membrane integrity are crucial metrics in any killing assay. On the left is a heat map demonstrating the count of live targets at 24 hours. The light to dark color shading indicates a low to high metric result in the heat map. On the right, for your reference, is the culture treatment plate map to see the correlating results. Of note, the excluded wells marked with an X are not shown as these wells did not contain target cells. Displayed here are the count of live targets and target membrane integrity results in heat map configurations for each time point. You can see, outlined in turquoise, the different stimulation treatments did not decrease target cell count or membrane integrity at any of the time points on targets alone. You will see that the number of live targets in co-cultures, outlined in yellow, does start dropping, slightly at 4 hours, but more pronounced at 24, 48, and 72 hours. And you can see that the positive bite has a more profound killing due to the bite itself bringing the targets and effectors into close proximity of one another. You will also notice while using the Dynabeads as the T cell activator, this demonstrates a nice dose response in both metrics, although the visualization by heat map is faint at 4 hours. Shown here, again using heat map configurations, are the percent activated CD8 positive T cells and percent exhausted CD8 positive T cells. At 4 hours, there's little expression, but by 24 hours, activation and exhaustion markers are expressed in both monocultures of effectors alone and co-cultures with each positive activation method. This is outlined in yellow. After 24 hours, we can visually see CD25 and PD1 expression occurring, and more specifically, we see a dose response when Dynabeads are used compared to the positive bite. Similar results were seen for the CD8 negative T cells, but the data is not shown here. Interferon gamma is an inflammation marker, and the presence of granzyme B indicates killing is in process. For each donor series, the yellow line represents 4 hours, turquoise is 24 hours, magenta is 48, and dark purple is 72 hours. Here are the 3 different donors and the measured amounts of interferon gamma and granzyme B with varying positive bite or Dynabeads concentration. Interferon gamma secretion is generally higher with Dynabead activation compared to the positive bite, while granzyme B secretion is higher with the positive bite compared to the Dynabeads. Granzyme B levels have a notable increase at 48 hours for each donor, and this corresponds to what was seen in previous heat map slide indicating the decrease of live cells and decrease in target membrane integrity. Also, a nice visualization of dose response is seen at the later time points. And outlined in blue, you can see that donor 3 secretion levels are quite different from donors 1 and 2. This poster was put together to describe a simple T-cell mediated killing assay workflow and to highlight the key features of the IQ Human T-cell killing kit and IQ Foresight software. This assay kit requires minimal sample volume and the short 3-hour assay time from start to finish, getting fast results. This kit can facilitate the screening of small molecules, biologics, or engineered cells in culture to determine T-cell mediated killing. These tools can be easily applied to profiling assays of cell therapeutics geared toward a more personalized medicine approach. Thank you.